thyroid function, you already know that it supports growth and development, especially of the embryo and brain. I'll give you some pictures that demonstrate that quite clearly. It helps regulate internal thermostasis, particularly in the young who contain brown fat, which is a real target of a lot of the thyroid hormones. And it helps maintain metabolic energy balance by increasing thyroid hormone would increase the number of mitochondria and the enzymes in electron transport chain, and also increases the number of sodium, potassium, ATPase ion pumps that are present in cell membranes. Okay. It's generally excitatory for normal cellular functions, including heart muscle. So if you have a thyroid problem, it often will show up as well in heart function. So it can be lethal if it's not maintained as normal. Thyroid turns out to be one of the major places in the endocrine system where disease occurs, where malfunctions occur. It, along with the pancreas, are probably the biggest sources of endocrine diseases and disorders. The big thing to notice in all of these instances, as we talk about hypothyroidism, too little thyroid hormone being produced, hyperthyroidism, too much thyroid hormone being produced. In both of these instances, there's between a 6 or 7 and an 8-fold higher incidence of those conditions in females than there are in males. And nobody knows why. Okay. At least not yet. There's iodine deficiency disorders. So you get endemic goiter, situations where iodine is deficient in the diet and causes inadequate production of thyroid hormone. As a result of that low thyroid hormone production, particularly in the mother, you get endemic cretinism. That is a neurodegenerative condition in offspring who have been deprived of thyroid hormone or iodine during development. And this is when this occurs, it's a lifelong problem. Endemic goiter can sometimes be corrected. There are many sources for this. Some of it I mentioned are iodine deficiency, but you can also get iodine deficiency in situations where the diet also contains compounds that block the incorporation of iodine into thyroid hormone. And those goitrogens are found in some species of yam, in other plants of the nightshade family, some of which are consumed quite broadly. But you can get things in the diet that will block iodine incorporation, the formation of thyroid hormone, even when there's sufficient iodine in the diet. You also have autoimmune type conditions like Hashimoto's thyroiditis, which is destructive of thyroid tissue and therefore suppresses thyroid hormone production. Hyperthyroidism is the other end of the spectrum, and you have topping the list Graves' disease, which is autoimmune thyrotoxicosis, which is the production of an antibody against the thyroid hormone receptor that stimulates that receptor, essentially takes the place of TSH, rather than blocking the receptor. So you end up with an antibody which is not under control, feedback control by the thyroid system, stimulating the thyroid and overproducing thyroid hormone. You also see thyrotoxicosis of pregnancy, which is often an imbalance of the production of the thyroid-binding globulin from the liver under the influence of estrogen, and the amount of thyroid hormone being produced by the thyroid gland. And then you get into multinodular goiter, which is toxic. That is, parts of the thyroid grow and overproduce thyroid hormone in what seems to be an inappropriate manner, and throws the individual into a hyperthyroid condition. And then there are the neoplasias, where you get benign enlargement or overt malignancy. Okay. And notice that thyroid neoplasia is the most common endocrine neoplasm. Thyroid anatomy, you should all go home and sit and feel your thyroids, okay? You'd be surprised as how many people walk into my office and walk out with a recommendation to see a physician. Okay. Pardon? This is more common than you might think, enlargement. This is a dissection showing you the two lobes of the tissue. Embedded in the tissue of the thyroid are the parathyroid glands, which have nothing to do with thyroid hormone production. They make parathyroid hormone, which is involved in calcium and bone metabolism. And then there are cells called clear cells, which exist in the in between what are called thyroid follicles, and those cells produce calcitonin, which is a counterbalance to parathyroid hormone, and again is involved in calcium metabolism and bone metabolism, not in control of thermogenesis and similar activities by the thyroid. This thyroid tissue, if you cut it through, you're going to see these open areas. This is the material inside is the product of these cells that line these follicles. There's a basement membrane around the outside that separates these spaces off from the vascular area. So this is an avascular space, and the protein there is called thyroglobulin. If I recall correctly, it's greater than 800,000 molecular weight on average. But you'll see that's probably a guess more than anything else because of the way this is both produced and broken down. So this is, because this is a storage area, and because the material here is iodinated, this is where a lot of the ingested iodine ends up being stored until needed in most mammals. In fact, in most animals that possess a thyroid of this type. And it is a means whereby animals can get away from the source of dietary iodine for periods, long periods of time, and not go into a hypothyroid state. You've seen this before, and the main thing to remember is that the key part of this feedback loop is TSH, or thyrotropin, from the thyrotrope, stimulating thyroid epithelial cells, the ones that make thyroxine and triiodothyronine, and those two hormones are what are the main negative feedback controlling thyroid production. The stuff up here is secondary controls. Now, a little more chemistry. Tyrosine. Okay, so here's your amino acid tyrosine. You learned all about it in basic biology or basic biochemistry. It's a side chain with a methylene group here and an aromatic ring, benzene-like ring, on which is attached an OH group. This OH group, in conjunction with a benzene ring, is called a phenolic ring. And that phenolic ring helps diffuse, or I should say distribute, the electrons from the oxygen over this ring and causes this H group to be more acidic. It acts like an acid group and comes off in alkaline pH. So above about pH 8, it would be ionized. Now, this grouping is what you normally find as thyroglobulin is being made. So it's made in the same way most proteins are. There's a transcript. It's spliced into a messenger RNA, a mature messenger RNA, translated into protein, normal amino acids in it. As it's being transported through the cytoplasm of the thyroid epithelial cells and into that thyroid colloid space, there is a reaction taking place at or near peroxisomes, which are modified lysosomes. And those lysosomes generate activated iodine species that allow iodination of these positions on this molecule, on the tyrosine molecule. Once you have that iodinated, you also get the possibility of shifting this grouping, this end grouping with the iodines on it, over to another molecule. And when you do, you create, depending on whether you've got two iodines on the grouping or one, you can create thyroxine, which is tetraiodothyronine, with 4 iodine atoms, or you can get triiodothyronine, which is going to be this molecule with 2 atoms of iodine on the inner ring and 1 on the outer ring. This is actually the hormone that binds to the thyroid receptors in most target cells. In fact, all target cells. This is the active hormone. This is the major product in circulation, and there's a conversion, a removal of one of these atoms in the target cells that allow this then to bind to the thyroid receptor. There, you can get formation of other molecules like this one called reverse T3, where you've removed one inner atom from thyroxine, or you've coupled a monoiodotyrosine to the diiodophenolic group, and you get something called reverse T3. And notice, even though what seems to be a small shift, this is inactive, whereas this is active. All of that is occurring in these epithelial cells surrounding the thyroid colloid and inside the basement membrane surrounding thyroid follicles. You have blood vessels outside bringing in nutrients, glucose, iodine, oxygen, as well as hormones, and carrying the binding globulins that will carry away the final hormones. The pathways that are key here are the generation of peroxide and the peroxidase activity here. So you form it and you break it down, and in breaking it down, you activate the iodine that can then couple with the thyroglobulin that's been synthesized. So this is d-iodothyroglobulin, and it's becoming iodinated as it's passing through the cytoplasm and coming in contact with the products of iodine activation generated by the peroxidase generating system. And this is called a pentose cycle, or the pentose shunt. So it takes glucose, generates reducing equivalents, and then these are used to form hydrogen peroxide, which then breaks down into two radicals, HO dot radicals, that are in fact involved in this chemistry. Okay. Then you have formation of these more complex amino acids out here in the, either at the surface of the cell here or in the thyroglobulin, or in the thyroid space proper, to form the more complicated amino acids. But they're still tethered in the amino acid chain. And it's really only after this is phagocytosed by these same cells and broken down by secondary lysosomes that you get release of T3 and T4 along with other iodotyrosines. The T3 and T4 then going into circulation, the other iodines being broken down by deiodinases into iodine, which is recycled, as well as amino acids. So this system works at a basal level normally, and when TSH stimulates the cyclic AMP and protein kinase A cascade, you get activation of transcription of thyroglobulin and activation, stimulation of glucose and iodine uptake and incorporation, as well as stimulation of phagocytosis. So it's going in both directions at the same time. Thyroid hormone transport, there's about 100 times more thyroid hormone in circulation than there is T3. It's bound by several major binding globulins. One is thyroid binding globulin, which sops up most of these. There's also some transported by a protein called transthyretin, and another one, albumin. The albumin is a low affinity, but there's so much of it that it still plays a role here. And then there's draining by receptors and by metabolism. This is just showing you the distribution of these various forms. There's of thyroid hormone among the various binding globulins. Let me go back. So these are micromoles in circulation of the binding globulin, and then percentage occupancy by T4. And about 30% of it is TBG, 2% of it transthyretin, and less than 1% to albumin. And then you have some of these other things about turnover rate. But the free concentration of the hormones are very, very low. Most is tied up with these proteins. This is breakdown of thyroid hormones. Thyroxine can be glycosidated, sulfated, a lot like steroids, and using the liver enzymes. You can take off the amine group, you can take off the carboxy group, and then break it down that way. You can remove the iodines at the 2 position, at the 1 position, using enzymes 1 or 2 or 3, and removing the 5' iodine or the 5 iodine. 5' is the outer ring, 5 is the inner ring. And then you get about half of it going toward T3 and reverse T3. You get more deiodination, and notice this is the same enzyme over here. This is the same enzyme over here. And you're going to diiodothyronine and then sulfation and breakdown finally by removal of all the iodine to thyronine. This is just showing you the affinity of these enzymes, or selectivity of these enzymes for the substrates. So you're getting, this is the Km, the maximal rate of activity of the enzyme at a given concentration of hormone. So the lower this is, the more effective it is on this molecule. So T4 is mainly broken down by these, reverse T3 by this one, and these only minor amounts broken down that way. There's links to other endocrine axes. You've seen that before. Somatostatin plays a role in controlling prolactin and controlling growth hormone, also seems to play some role in controlling thyroid hormone. TRH, although originally isolated and named by people who were working on the thyroid system, find that this also turns out to be involved in control of prolactin. So if you have low thyroid levels, you can get elevations of TRH and potentially involve overproduction of prolactin. And these are just indirect links to all the other steroids. So how does thyroid hormone act? First of all, the molecules I showed you, are those charged molecules, uncharged molecules, big molecules, small molecules? What are they? How much charge? Okay. We normally check the charge at neutral pH. Okay. Any guesses? Why do you say charged? Pardon? The OH group at neutral pH, remember I said it ionized at alkaline pH. So at neutral pH, that OH group is intact. No charge there. Well, there are amino acids, so there's going to be a partial positive charge on the amine group and a partial negative charge on the OH group. What's the net charge? Close to 0, very close to 0. Okay. Now, those iodine atoms weigh about 127 atomic units each, which is equivalent to or more than the rest of the molecule put together. Carbon is 12, oxygen is 16, hydrogen 1. Okay? So those things are sticking out like big balloons, and they are what dominate the chemistry of those molecules. And they tend to be lipophilic. They behave a lot like steroids, a lot like lipids. Their passage through cell membranes is in part passive. So passive diffusion because of the lipophilic character. they also co-opt amino acid transport systems that would normally bring in tyrosine, would bring in phenylalanine, would bring in a lot of those big neutral amino acids that tend to be kind of hydrophobic in character. And so it's the passage into the cell is both by passive diffusion through the membrane and via facilitated diffusion using those other amino acid transport systems. Once it gets into the cell, okay, given that it's a relatively neutral molecule, is it a big molecule or small molecule? I told you the atoms were big. 127, 127, 127, 127. Okay. What's 4 times 127? Quick. How about 528? Okay. Plus another 100 or thereabouts for the rest of the molecule. So you're up 600, 700. Is 700 molecular weight a big molecule? What's big? What things are big? Proteins. Lots of the macromolecules, proteins, DNAs, RNAs, thousands of molecular weight. What's something else that's small? Neurotransmitters. What else? Nucleotides. What else? Steroids. What else? Ions. What else? How about, is cyclic AMP big or small? Small. How about cyclic GMP? Small. Okay. You get the point. Okay. It's the smaller end of the spectrum. So yes, that may be big according to amino acid sizes, but it's small relative to a lot of other macromolecules. So what kind of a receptor does it normally use? Is it a surface receptor? Somebody said it. A nuclear receptor. Okay? I heard you as well. A nuclear receptor. Okay? And in fact, there are multiple nuclear receptors for not T4, but for T3. There are 4 functional intranuclear T3 receptors. That should be, beta, that should be alpha 1, beta 2, 1 and 2 and 3, and 1 non-functional receptor, which is alpha 2. There's expression varies across tissues and developmental stage. So you've got 4 related receptors, alpha 1, alpha 2, beta 1, and beta 2. Notice that alpha 2 doesn't bind the hormone. So if it's expressed, what's true? If it were expressed, so this may be a pseudogene, but if it were expressed, what would happen in the presence of that receptor? What happens with these receptors? What do they normally do? They bind the hormone, and then what? Cascade? Do you get a cascade with nuclear receptors? Pardon? You alter transcription. So if you've got a receptor and it's being expressed, but doesn't need hormone to act, what can happen? What can happen? What? You activate transcription without hormone binding. So this becomes non-regulated. That becomes a constitutive activator. So this better be a pseudogene, or you've got a constitutively activated system. Notice that all of the rest of these have DNA binding, so they can activate transcription, but they also bind T3. And as I said, they differ as to where they're expressed and when they're expressed. Pregnancy, I mentioned, activates the thyroid axis in part because you increase the thyroid binding globulin, you increase plasma volume, but you also increase TSH, and, HCG, and this can act, cross-react with TSH receptors and stimulate thyroid function. You've got D3 expression in placenta, which is the deiodinase. So you can clear mom's thyroid hormone, or you can clear fetal thyroid hormone by placental deiodinase. You got renal clearance of the hormone increases. Fetal synthesis in late second and third trimester is what tends to stimulate oxygen consumption, increase basal metabolic rate, and ultimately end up stimulating the birth process. Here's maternal axis impact, maternal thyroid axis, increased T4 production, increased total T3 and T4, total pool, cardiac output, free T4, decreases the basal TSH, but it increases the requirement for iodine. So during pregnancy, part of the reason you get thyroid problems is because of this increase in iodine requirement. So she has to make sure she's taking in enough, or she's in an environment where she can recycle the iodine she has. I'm going to take you on a trip. You guys ready for a trip?
Where are we going?
We're going to New Guinea. Okay. Anybody else been to New Guinea? No? Okay. I'm going to go to New Guinea, where we did some a population study that ended up involving looking at the thyroid axis. We were looking at determinants of fertility in a population that was non-Western, was non-contracepting, and yet women who lived through their reproductive lifespan only had 3 to 4 children, which is way, way below what you get in a Western context in some, for instance, religious sects that maximize reproduction by wet nursing babies, so moms come back into reproductive condition quickly after birth, and they start by marrying young and continuing to have children kind of one after the other for years. And under those circumstances, the lowest numbers are 6, and then more likely you're up at 10, 11, 12 as the completed family size. 18 has been also recorded.
I think they're at 19.
Yeah, I think tops is around 30, and it has to do with multiple births. Okay. So where are the Gainch? They're in a little pocket up here in the New Guinea highlands. And we talked about the dagger of Australia pointed at the heart of New Guinea. And this kind of points almost at the area that we work in. And so they're up in the valleys up here. I'm getting you closer and closer. So it's a river valley up in the highlands. This is a malarial coastal floodplain. Here you're up around 3,000 to 6,000 feet. So you're off of the malarial floodplain. It's still tortuous terrain, and there's lots of populations living in almost isolation in these areas. This country is very interesting because of the activity. There's the Australian plate of the Earth sits about like this, comes up to about this midline. And then the Pacific plate is over here. And so you get a lot of tectonic activity. There's also some active volcanoes out here. And so this is a very active areas. The geology is rugged, and there's lots of isolation of populations. As a result, there's been developed about 800 different languages, distinct languages, not dialects, but distinct languages, in the peoples that populate the highlands of New Guinea. So they're often evolutionarily isolated groups with minimal contact to the outside world, definitely, and somewhat to the groups immediately around them. The Gainch were one of these, and their intriguing reproductive history. The women mature somewhere in the late teens to early 20s. They have their first child around age 25. And then about every 3 to 4 years, they would have another child. But it ends somewhere around age 35 to 40. And in part, it turns out that it's because of these guys. Okay. When we looked at the reproductive status of males, they develop late as well. So they mature by the late teens into the mid-20s, but because of cultural practices about how you form families, these guys go out and raise bride price. So they often migrate to the coast and come back around age 30 to find a bride and raise a family. When you look at their hormonal profiles, it turns out that their hormonal profile suggests a decrease in fertility by about age 35 to 40. So they have a very narrow window during which they are actually optimally fertile. So this is one population in which the men limit total fertility, okay, along with the wide spacing of childbirth. This woman is probably in her 50s or late 40s. But because of the labor and child rearing in rugged conditions, they age quite rapidly. This is just a description, a summary of some of what I've just told you.
It's been menopause is earlier too, so it's just a shorter.
Well, menopause isn't, we don't know because the numbers aren't there really to give us.
That one just said 46.
Yeah, that's best estimate at this point. Okay.
Which is younger than Western women, isn't it?
Not by much. It's maybe 5 years.
5 years makes a difference.
Pardon?
5 years makes a difference because it makes a difference on the front end.
Okay. But if you have an infertile male to deal with as a partner, even if your menopause is later or a little earlier,
then you're double screwed.
yeah, it's not going to increase fertility. So this is just hormonal levels of these guys relative to several other populations, and demonstrating that relative to the number of females ever married, the males are maturing late. This is what happens to prolactin levels. The prolactin for Western groups and a selected group of people in the laboratory where this was originally tested are pretty low, as are the Gainch males. But if you look at the Gainch females, this level of 24.2 would clinically, in the West, would be clinical evidence of hyperprolactinemia and would normally be, they'd be looking for tumors or, and anticipating that the reproductive cycles would be shut down. These women are cycling. No goiter. So there's prolonged intensive nursing that keeps prolactin high and ovulation suppressed. If you look at the age of the child versus serum prolactin, you're out well beyond 2 years before you get to what is considered the normal range for serum prolactin in non-lactating individuals. Okay, so this is just proportions to lactating. So it takes a long time before they come down. They're continuing to lactate for a prolonged period of time, but the time until their prolactin levels are low enough to allow to cycling again is around 2 years. So prolactin decrease during lactation was still clinically high, implying TRH might be high, TSH should be high, and T4 should be high unless iodine deficiency was present. The thyroid axis pathology might help explain low fertility. Was there any evidence? Well, this is a classical picture you've seen before of a hyaline goiter in an endemic iodine deficient area. We didn't see this. In fact, you saw very minimal enlargement of the thyroid glands in the population we were looking at. What we did see, however, were several cases of cretinism. This is a cretin. She's probably in the teens. And has a very limited capacity to do anything besides exist.
What's
A cretin is an individual who is born with iodine deficiency prior to birth and continuing after birth. Clinically, this used to be a problem in the Great Lakes and other areas where iodine stores were limited. What happens now, does anybody, I don't see her here today. Has anybody had children here other than me? Okay. Do you know, what do they do to the child within a couple of days after birth? Pardon? You didn't see her? Okay. Well, one of the things they do is what's called a heel stick. They'll do it very early on, but it's certainly within a couple days of birth. And what they're doing is they're collecting blood samples on pieces of paper, and they're going to test those for a variety of things. One of which is T4, one of which is TSH. They also look for metabolic diseases like phenylketonuria. The reason they do that is because most of those conditions can be treated if caught early enough. And it's become virtually almost worldwide where that screening has actually been done to prevent this kind of an occurrence. But in this population, there were at least a couple of them around, so we knew there was a thyroid problem. Yeah?
How long do those children live like that?
They'll probably die by the time they're 30.
Shakespeare told freedom was an insult.
So if you looked at thyroid hormone, TSH, there's pretty much normal in both men and women. There are a few above it indicating hypothyroidism. but the TSH looked pretty normal. The T4, total T4 levels, again, weren't that far off. Free thyroxine in serum, again, fell in the normal range. So why do we have cretins in the population, and why do we have potential thyroid elevations of prolactin that may be associated with thyroid problems? So the hormone levels look pretty normal. That's called euthyroid. Whoops, I'm back. What about the carrier proteins? Because one way you can get malfunctions are having the carrier protein levels be disrupted. Okay. And when you look at serum albumin, the one thing you note is that in women particularly, it's at the low end or below normal. This is coming about because the protein in the diet of these individuals is low. Okay. Largely vegetarian diet, some insects and arachnids being consumed, but very little in the way of protein, other protein. Serum prealbumin, which is transthyretin, also on the low end, probably associated, but that's both in men and women. Interestingly here, the serum albumin in men is higher than it is in women. And that probably has to do with access to protein in the diet. Okay. Well, when you looked at the total, you looked at the total thyroxine and the thyroxine binding globulin, the normal range is here. What you see is this in the Gynge populations. For the men, and for the women, and for pregnant women it's even more pronounced. And that is that you get an elevation of the thyroxine binding globulin, even in the normal range of thyroxine. So what does this do? Well, they have high TBG, but normal thyroxine, and it tends to be a compensation for low dietary protein and dietary iodine. It elevates iodine elevates TBG when other carrier proteins decline, and it prolongs thyroxine life and decreases iodine needs. So here we have a physiological adjustment to both low protein and low iodine in the diet that allows them to function in a relatively normal endocrine environment.
So the compensation for low dietary protein, that's so that it's increasing basal metabolic rate in order to compensate for that.
Yeah, remember this are a couple of things. TBG is present in about 1/1000th of the amount of albumin present in circulation. Okay, so this is a low abundance protein in circulation. Doesn't take a lot of protein to support production of that protein and maintain it in the normal range. If you've got low iodine, but you've got a lot of this will tie up T4 and T3 in circulation and maintain them in circulation for a more prolonged period of time because it keeps it away from the catabolic pathways that normally would clear it. So you've got that combination of things maintaining iodine and thyroxine levels toward the normal range in these individuals. And so they perform and function normally, but in a deficient environment. What this suggests, however, is that a problem arises if you give back iodine only. So many of the World Health Organization recommendations for treatment of iodine-deficient populations is to give iodinized salt or iodinized oil, okay? But in this circumstance, if you give those alone without giving protein, what do you get? Any guesses?
High thyroid production.
You get hyperthyroidism. you get thyrotoxicosis, okay? And that's exactly what they've seen in some other populations that they've treated, but nobody has really pointed this out before.
So you would need to increase protein levels as
You
well.
need to increase protein levels so these other binding globulins go up to stop